Thank you, Basanti. A good afternoon to all our audience. Over here are our participants. Thank you for taking your time to join us, be it in the morning or afternoon, or some of you will be in the evening. We would like to take this opportunity to introduce our speaker for this time round. We have Mr. Benoit Lemon, who has turned on his video. Hi. Good morning to you, Benoit. Yes.
Hello.
So Benoit Lemon has been with Thermo Fisher for more than 10 years. He is our bioprocessing application specialist for the floor model centrifugation. Benoit holds a master's degree in automation from the University of Sophia Antipolis, France. And with him, he brings a lot of experience because he has been working for more than 10 years in industrial downstream bioprocessing, focusing on vaccine and gene therapy purification. Some of the big customers he worked with in the designing of the bioprocessing downstream workflow with customers like Sanofi Pasteur and Pfizer. So without further ado, I would like to hand this time over to Benoit. If you would like to start your webinar on what are important considerations for centrifugation in the bioprocess workflow. So over to you, Benoit. Thank you.
Thank you, Anita. Thank you, Vasanti. Stop my video and we get started in a minute. All right. Good day, everyone. So, thanks, Anita and Vasanti, for the introduction. My name is Benoit Lemon, and I work for Thermo Fisher Scientific, part of the Centrifuge business unit, as an application person focusing on the bioprocess application. And today, during about 40 minutes, I will be covering the important consideration for the centrifugation in the bioprocess workflow. So there will be 3 sections in the presentation, and please feel free to use the chat to ask your questions, comments, and there will be about 15 minutes after the presentation to go through your through the questions. So first we will remind us the bioprocess application, and we'll be more specific on which typical application the centrifuge plays an important part. As a second session, we will touch the typical pain points of centrifugation in the bioprocess workflow, and we will discuss the important consideration around these points. And to finish on the section number 3, we will look at the specific centrifuge for the bioprocess application with Thermo Fisher Scientific. So let's get started with the section 1. We will talk about the upstream, the downstream, and the different steps of the workflow, which are part of the downstream, in which the centrifuge plays an important part, which are the harvest, the clarification, the purification, concentration, and polishing. Before we go into the detail, let's remember what is the bioprocess typical work. As all of us know, the bioprocess is to produce, to manufacture a therapeutic or biological product and usually the products are produced by a cell, by a living organism or by a living cells. Yeah? The cells are used at a factory. They are grown in incubator or fermenter or bioreactor. And then once they get to the size, the desired size, expansion volume, and the cells are producing the therapeutic, the biological products. Then this product is collected, is harvested, is clarified, and it's purified to remove all the contaminants and to obtain a pure biological product of interest. So the bioprocess is the workflow to obtain this product of interest for therapeutic means or other means. The upstream process is to grow the cells and make the cell produce the product of interest. Depending on the application, the product of interest may be intra cells or extra cells. The cells, they grow in a culture media. So sometimes the product of interest may be kept inside the cells, or sometimes the cell may release the product of interest outside in the culture media. The centrifuge are mainly not used during this expansion upstream process. At the end of the upstream, there is a vessel, maybe ranging from 1 liter up to 10,000 liters, depending on the size of the industry or the target of the product and the phase, if we are in development, in research, or in mass production. And we have cells, living cells in the culture media, and one or the other is containing the product of interest. Then the upstream is always followed by the downstream, and the goal of the downstream is to separate the biomass harvest, so to separate the cell from the culture media. So in case the cell is containing the product of interest, then the culture media, also called the supernatant, is discarded, and only the cells containing the product of interest are kept, or the way around, in case the product of interest is released out of the cell into the supernatant during the upstream. So the first step here is the separation of the biomass, the pellet, the cells from the supernatant. This is also called the harvest step. Then, if the cells are containing the product of interest, then the next step is to lysate the cell, first to resuspend the cell in a neutral buffer and to lysate the cell. So the product of interest contained inside the cell will then be released out into the suspension buffer. So at this stage, the cell will be debris or contaminant, and then the resuspension buffer containing the released product of interest will be clarified to remove the cell debris contaminants. It can be the way around if the supernatant is containing the product of interest from the harvest step, and the cells are removed, then this liquid will also go through clarification to remove dead cells, cell debris, and other micro-size contaminants. So this step is called the clarification of the liquid. So after the clarification, usually the cells and the cell debris and the micro-sized particles are all removed, and the liquid is containing the product of interest as well as some other nano-size particles, contaminants. For example, if the DNA molecule is the product of interest, it could be very well possible that the cell also released protein, RNA, and other type of molecules, meaning there should be a separation of the molecule of interest from other contaminant molecule. And this is called the purification, the concentration, and the polishing of the product of interest to obtain during the final formulation a pure product for packing and going to the commercial. This chart is showing you the typical curve for volume upstream, starting with some few milliliters and expanding on the upstream from 1 liter up to maybe 10,000 liters, followed by the downstream. And you see in a red color the harvest and the collection phase. So the harvest is the separation and the collection of the product of interest from the culture media, followed by the clarification, which is the removing of the liquid substance from the product of interest, and finished by the purification, concentration, and the polishing of the product of interest before the bulk storage and the final fill. I have a question for you. I would like to know, with your daily work, if you are using centrifuge for the different harvest, or if you are using centrifuge for harvest, or clarification, or purification in your bioprocess workflow. So you can answer the question to everyone using the chat box that should be displayed on the screen. So let's see if it works, and give me a few answers. So if you guys are using centrifuge for harvest, for clarification, or for purification. All right, so I don't see much answers coming. So I don't know if the chat works, or or if something else. So I just carry on, and feel free to answer whenever you wish. Thank you. So the next slides are showing some typical application workflow for bioprocess using the centrifuge at some of the different steps. This is the example of the cell therapy manufacturing. In this case, the cells are collected from a patient. They are mixed with some media for cultivation, and they are harvested and concentrated using a centrifuge. Usually the whole cell is the product of interest, and the centrifugation should be very gentle, not to damage the fragile and very delicate and important cells. So usually we are using between 500 up to 1000 G-force. The size of the whole cell is quite big compared to the contaminant and the media. So the separation is quite easy, and there should be no shear effect, only gentle force to be delicate with the cells. At the end of the centrifugation, the whole cell are harvested, and the supernatant media is discarded. Then the cell are resuspended with buffer, some buffer exchange, and then some following steps, usually not requiring centrifuges. In case of gene therapy manufacturing, usually this is a multi-steps process, and the centrifuges, they are used at different steps, and different type of centrifuges are also used. Usually the process starts with the preparation of the DNA vector or the DNA plasmid. The DNA plasmid is usually grown on bacteria, on E. coli or microbial cells as a separate process, and centrifuge are used to separate the microcell from the microbial cell from the supernatant. This is about 1 to 2 micro size cells, and they are very robust, and usually speed of centrifuge between 5,000 to 10,000 G is used to pellet the micro cell and to harvest the pellet and discard the supernatant. The microbial cell will then be lysate to release the DNA plasmid, which will be collected, and then this DNA plasmid will be used as a vector to transfect a cell, typically HEK 293 cells, grown on the bio reactor. The cells grown on bioreactor are transfected by the DNA plasmid vector to modify the DNA of the cells, and usually the cells are releasing now the viral vector on the culture media, and then the supernatant is harvested, usually with the force between 500 to 5,000 G. Again, to be delicate with the cells. We don't want cell debris to contaminate the supernatant, so as much as possible the cells will be kept intact and left on the side for waste, and then the supernatant containing the viral vector will be harvested and clarified to remove any remaining debris. This is requiring a little bit higher G-force, maybe 10 to 20,000 G. And then going to the purification. This could be some chromatography system, and combined with the centrifugation, typically a gradient centrifugation to separate the full from the empty capsid. The capsid, they're very small size, and some capsid contain the DNA cassette information, some don't when they replicate. And a possible method to separate the full from the empty being slightly different weights. One possible method is to use the zonal gradient separation at 100,000 up to 200,000 G-force. This is the typical workflow for manufacturing the monoclonal antibodies. And again, after the cell cultivation, the supernatant is harvested at a speed between 500 to 5,000 G to remove the cells, and then the clarification to remove the cell debris and obtain a clarified liquid with the product of interest, and other contaminants are removed with the purification before the final steps. In case of vaccine manufacturing, this is the example of using the egg-based for manufacturing the vaccine. Centrifuge is very popular at also different step of the workflow. The clarification is to remove the debris of the egg and anything not the allantoic fluid containing the virus. So this will be usually a big centrifuge, 10 to 20,000 G-force to separate cell debris from the allantoic fluid. Then the allantoic fluid containing the virus are processed through a zonal gradient ultra centrifuge and about 100,000 G-force. And the virus, they will be sorted by size and density in the gradient, and a specific fraction will be recovered for the targeted size virus. Then usually followed by the inactivation and the virus split phase, and then one more ultra centrifugation, 100,000 G-force, is used to isolate a specific part of the virus, making this liquid solid separation and usually harvesting the antigen on the supernatant, followed by the final formulation and packing. This mRNA viral vaccine, typically the COVID vaccine that we all hear on the news nowadays. This is also using the centrifuge as the step to prepare the DNA vector, almost similar to the gene therapy manufacturing. So growing microbial cell, producing the DNA plasmid, the DNA vector, and using the centrifuge, again 5 to 10,000 G, to harvest the microbial cell and lysate the DNA vector, and then again using the centrifuge for the harvest and the clarification step after the cell cultivation and transfection, and followed by the purification and the final formulation. This is the last example I have for you guys. This is the cell-free protein expression manufacturing, and in this case the cells are evacuated on the density gradient. The vacuole is removed from the cell, maybe 5 to 10,000 G, followed by the lysate, and then the clarification is also using some centrifuge, followed by the purification and the following step. I'd be interested to know what type of application you guys are using with the centrifuge, with the different steps. So feel free to use the chat and tell me if you do vaccine, if you do mRNA vaccine or other type of products. And when you answer the chat, just carry on the slides. So we will now touch some typical pain points using the centrifuge in the bioprocess workflow and see how they can be addressed for the centrifugation at the different step of the workflow. So we will be talking about the contamination control, about the scalability, about the compliance, about the flexibility, and about the reliability. The contamination control. So I think from what we hear, the contamination is the number one pain point for the user because they can endanger all type of cell culture. And they can ruin all the work and this can happen at any step of the workflow. This can void the commercial release of the product and maybe have impact on the reputation of the company. And of course, the best way to prevent the contamination is to have a strategy based on prevention and of course the eradication of the contamination where it does occur. In case of the centrifugation, the good practice is to avoid the contamination, is to ensure that the containers are clean and sterilized. Meaning performing some validation, cleaning validation, sterilization validation, to ensure there is no contaminant organism, or to use a single-use sterilized container. So the process is completely closed. There is no cleaning and sterilization involved, and the container, the tubing, the connection, they are all everything comes sterilized, ready to use, and reducing and this is reducing the risk of contamination. And of course, manipulate the container in a clean air environment when loading and emptying the system. The container, the system, or to use the closed system from the bioreactor to the container, centrifuge, and through the workflow. In case of zonal centrifugation or continuous flow, the goal is to ensure the cleanability and drainability of the product path, so no contaminants. And it's possible to use either the clean out of place or the clean in place for the product path and ensure the sterility by chemical or steam performing validation or using again a single-use sterilized closed system assembly. And the other method to prevent contaminant is to prevent from potential leaks on the continuous flow system. And the graph is showing pressure differential control, having the stream of products at the highest pressure, then the utility pressure will ensure, in case of leaks or seal defects, that the product is safe because of the highest pressure. So this table is showing just a quick summary of what could be the contamination control with the centrifuge used at the harvest and the clarification step of the bioprocess workflow. One can use a sterilized single-use container. Usually it's shipped with a sterility certificate, ready to use, no post-use cleaning required. And of course, one can use the cleaning service for the containers, either outsource the cleaning and sterilization of the container or to do everything in-house. And in case of the purification, concentration, polishing, if the continuous flow machine is used, then it's possible to use an automated system called the integrated process control to maintain the positive pressure through the seal and to avoid to have the positive pressure cascade. Of course, be careful the coolant, sanitization, the clean in place, the steam in place ability of the design to ensure all the lines are clean and free of contaminants, and monitoring the level of the different liquid and coolant lubricants to ensure no leakage and to be able to prevent, and the possibility to use some HEPA filter between the chamber of the centrifuge and the outside of the centrifuge. When we speak to user, the second pain point we hear is the scalability. There might be a challenge or pain points how to scale up, how to scale down, and how to create a small scale model for the centrifuge. So the scalability is very important because you guys are developing the process on a small scale, but always keep in mind the scaling up, keeping the same parameters of the process. So one common question is, how do we scale up with the centrifuge? And then once the scale up factory is ready and running, one would like to create a scale down model in case process optimization is required, and then change some parameters on small scale, on the scale down model, to test and validate before implementing the change on the large scale factory. So the scalability with the centrifugation works rather with the scale out and scale in, rather than the scale up and scale down. So the scale out is meaning increasing the quantity of machine or the number of containers or the volume of containers, but always keeping the same separation characteristic. So the separation characteristic is the G-force and the K factor. So once you keep the G-force and the K factor equivalent, and you can increase the size, the scale, by increasing the numbers of containers, or volume of containers, or quantity of machine. The advantage is that the small scale will be very, very equivalent to the large scale. In case of zonal centrifugation or continuous flow, then it's a matter of changing the flow rate and keeping the G-force and the K-factor equivalent. So here you see some example of scalability for the harvest and the clarification. This can be answered by different type, different size of container, different quantity of containers, and that's it. Different volume and different quantity of containers. And when looking at the continuous flow system, you see the rotor or the insert can have different volume, so meaning changing the flow rate, changing the scale of the process, but always keeping the same separation characteristics with the with the same radius, the same minimum radius and maximum radius, and the same G-force. The compliance is always key in mind when considering the bioprocess application. The compliance, of course, is to ensure the quality and the regulatory and control the strategy for the data integrity, the traceability, the validation, and the certification. For the compliance for the equipment means to comply with the safety standard in place in the country, as well as the traceability of the sample. Usually the software or the equipment will do the batch data reporting and the audit trail to track all the work and all the samples. The connectivity will allow the management of the user, the time synchronization with the network for the batch data reporting, as well as the data archiving and the system backup. And of course, the ISO certification. For the product contact containers, one should look at the certification. For example, the USP Class 6 approved, ensure about the extractable and the leachable, that the products are manufactured in some ISO 7 clean room, and all the data are available and listed on some certificates, and that the product comes sterilized with the certificate, and of course with the ISO certification. So here is shown an example of certification report for product contact container, listing the different guidelines and compliance confirmation. So as a summary for compliance, using the centrifuge for the harvest or clarification or purification step, one should look at the material of the container to ensure the chemical compatibility with the process, and the compatibility with the temperature, as well as the certification like FDA, USP Class 6, TBE, BSC-free, extractable and leachable, and the sterility, and ensuring that the product meets the safety standard, that the qualification services are available, and the software for traceability as well, and the connectivity of the device is also available. Looking at the flexibility of centrifuge for the bioprocess workflow. So the flexibility is to ensure the equipment can be operated at different scale for different products and be capable to produce multiple different products. This is very useful in case of new projects or clinical trials, when there is a change needed in the target application or the size of application. So for centrifugation, the flexibility comes with the range of different rotor, different type of machine, and different size of container. And of course, the full flexibility is selectable on the user interface of the equipment, having the different G-force, different separation time, temperature, acceleration and deceleration stop. And in case of this continuous flow centrifuge, the flexibility comes with the automation device on which is possible to program the recipe in many different manner. Would like to finish this section with the reliability points. So the reliability is to produce a constant result and to ensure the most uptime of the equipment. For the consumable, it's to ensure the good cleaning practice, the good decontamination practice, inspection, and the lifetime usage. For the equipment, the balancing of the rotor, the cleaning of the equipment, and the maintenance of the equipment. I would like to finish the presentation a few minutes looking at the centrifuge for bioprocess with the Thermo Fisher, and then I'll be happy to answer the question on the chat room. So few more minutes to finish the presentation, and time to prepare questions if you guys have any. In the Thermo Fisher for bioprocess centrifuge, we have a complete offering of machine called the BIOS 16 for the harvesting, the BIOS A, the Lynx 4000, the Lynx 6000 for the harvesting, and for the clarification and the harvesting, and the model Sorvol WX Plus, CC40 SNX, and CC40 NX for the purification, concentration, and polishing. The BIOS 16 is the harvest machine to expand your batch production capacity and the cell recovery without a capital investment. The BIOS A machine is providing a higher G-force and capacity to enhance your batch production capacity. The Lynx super speed centrifuge is fast and efficient separation of cell culture from the bioreactor at high G-force, providing a simple and safety and reliable way to clarify your liquid. The Sorvol WX Ultra Centrifuge is up to 100,000 RPM. This is to separate nano-sized particles in small footprints, mainly in the R&D work. And the Sorvol CC40 is the scale-up machine of the WX with the full scalability of the product line for the large scale purification up to 100 liters of the nano-sized particle for vaccine or viral vector purification. This table is showing a summary of the different containers, volume, maximum speed, maximum G-force, and typical application for the different centrifuge for bioprocess within Thermo Fisher. So this is offering the full range from the 1 liter bottle up to the 8 liters continuous flow zonal rotor from as minimum as 500 G-force up to 120,000 G-force, addressing the different steps of the bioprocess workflow, being the harvest step, the clarification step, and the purification step of the different application, different type of cells for separating mammalian cell, yeast cell, bacteria cells, protein, virus, and other type of micro-size and nano-size particles. Please visit thermofisher.com/flex4whatnext, and you will find plenty of material, animation, and information specific on centrifuge for bioprocess. So thank you all for your attention, and I think we can start the Q&A session now for the remaining 10 minutes. Just give me 1 minute. All right, I see the first question. First question I see is, what is the preferred process volume when harvesting within the Thermo Scientific Solval BIOS 16 centrifuge? So the BIOS 16 centrifuge is a batch centrifuge. It's possible to perform up to 16 liters per batch. So typically the user are using up to, I would say, 50 to 100 liters divided in 16 liters batch to be processed with the BIOS 16 centrifuge. The next question, what is the typical application for the Thermo Scientific Solval CC40NX ultra speed centrifuge, and how is it different from the other centrifuge? So the CC40 is this large scale vertical rotor centrifuge up to 120,000 G-force. So it's used to separate the nano-sized particle, and it's used with the density gradient, liquid-liquid separation, or the pelting, liquid-solid separation. And because the rotor is vertical, it's possible to, and continuous flow, it's possible to automatize the recipe and to provide a closed system for very delicate material, very sensible to contamination. So we can have a free contamination, no handling of the sample. The machine and the integrated process control can do the complete work of loading, separating, unloading, analyzing, and sorting the fraction in the container. Everything automated, everything closed system. I see the next question from Leo. Is the separation and purification process, in the separation and the purification process, the difference between centrifuge, chromatography, ultracentrifuge, ultrafiltration technology, could they replace each other? Okay, so thank you for the question. In the purification, there is 3 techniques. The separation by density with the centrifuge, the separation with the chemical or affinity or ion exchange, which is the chromatography, or the separation by size, the ultrafiltration. And so these 3 technologies, they use a different physics to make the separation. And depending on the process, they can replace each other or not. It's really up to the science. And I will say the centrifuge is capable to give the best results because it's independent of the size and independent of the outside of the particles. It makes the separation based on density. And so sometimes it's one type of separation and sometimes it's a mix of the different separation. Like the gene therapy application is a mix of the 3 techniques. The first technique, the chromatography, to separate the viral vector with the capture select based on the affinity. To isolate the full and empty capsid from the contaminant, then the centrifuge to isolate the full capsid from the empty capsid in the density gradient, and then the IEX, the ion exchange, to remove the density gradient from the full capsid. So different techniques and different strategy based on the product. The next question is a traceability software available to support the GMP application? Yes, in case of the Thermo Fisher centrifuge, the different machine comes with different software, and the software will support the full GMP compliance for your bioprocess application with the user management, the sample management, the batch data, and the audit trail. I see another question just earlier in the chat. What kind of centrifuge you will select after T-cell clonal expansion for cell therapy in cell culture bag? And what are the most common cell culture bag in terms of size you have seen so far? I don't know the answer for this question. I will copy and make some study and reply to the host of the webinar a little bit later. Thank you for the question. Here I see a question. Could you please elaborate more about centrifuge cleaning and validation in the clean room? Again, I will do some research and I can provide a detailed answer to you, Rose Quinn. Also replying to the host of the webinar a little later. Okay, I think this is it for the questions. We have a few minutes remaining. Back to you, Anita.
Thank you, Benoit. With this, I think we have come to the end of the seminar. Thank you, everybody, for attending. For those who have asked questions where Benoit will do more research, we will get back to you. I believe you registered with your email. We will respond to you via email to your question asked. So with this, I thank you very much for your time and all the participants for your time in attending this webinar. Thank you, and we will see you again soon. Bye.